So I am Pankaj Gautam from IIT Ropar. I am working with Dr. Sudipta Kumar Sinha, and the title of my today's talk is Anticipating Response Function in Gene Regulatory Networks. So the outline of my talk will be, first of all, I'll be giving an introduction to gene expression, then what are the objectives and of the proposed problems, then model system, uh, which we have applied on gene regulatory networks, and followed by its application on bacterial system, and then I'll be concluding it. So what is gene regulation? Gene regulation refers to the term that controls the gene expression. So we have DNA, which gets converted to mRNA using this RNA polymerase. So it binds on the promoter region and unzip this DNA and forms the mRNA layer. This process is called transcription. And from this mRNA, organelle ribosome will bind and it will form the protein layer, which is called translation. So what are the regulatory region for this gene expression, where the regulation happens? So there are a special type of proteins which are called transcription factor. They binds on DNA, and that's how they control. And there is strength of input signal that is responsible. So this input signal can be anything externally added, any biomolecule or compound, for example, insulin, or it can be related to phosphorylation or acetylation also. And there can be self-assembly of this transcription factor also. For example, in this case here, it is forming tetrameric species. And it also depends upon the physical state of DNA, whether DNA is in a looped form or in unlooped form. For example, it is forming loop here. So everything was going fine. So what are the objective and problems that we have figured out here? So we know that in a healthy cell, genetic response function is ordered. Even though cell is a very noisy system, still a healthy cell has a ordered genetic response. And this response function is somehow linked with the biomolecular interactions that are happening at the gene level. And correlation between this mechanism and response function is actually still not known with clarity till date. So if we are able to understand this mechanism, so then how it will help us? It will help us in reducing the experimental efforts by forecasting the outcomes and in the development of disease-specific molecular therapies. So then we made an objective for us. What is our objective? That we want to study this relation between input signal and response function by incorporating various biophysical events. For example, the one which I have discussed previously. So now we are moving towards formulating our system. So our system setup, we consider two assumptions. For example, average behavior of a network is invariant over time. Means we are dealing in equilibrium regime. And we are also considering that binding kinetics is much faster than the other cellular processes, for example, cell growth. So this is our schematic picture that we have considered. For example, we know that consider this as a nucleosome where the DNA is coiled around histone protein. So if we zoom into it, so then at genetic level, various biophysical events or biomolecular reactions are happening. So consider this green one as RNA polymerase, this brown as transcription factor, and magenta as ligand. So in the next coming slides, colors will be remaining the same and will have the same meaning. So then DNA can be in loop form also or in unlooped form also. So now what we have done, we have assumed that let us map this DNA as a one-dimensional array of DNA, where these beads, which are actually representing biomolecules, they are binding and interacting and are following the various genomic laws. So then there could be so these biomolecules can be placed anywhere depending upon these are like randomly placed, and they will be responsible for forming the loops also. So these are the two microstates we have shown here. For example, microstate 1 and microstate 2, and depending upon the system, we can have multiple microstates. So then cell is an example of open system. So that's why we want to deal with it in grand canonical by using grand canonical partition function, which is having a relation with the zeta term, which we are calling as promoter partition function. Promoter partition function, why we are calling it? Because it is the partition function that is related with the promoter. So so now all this problem is actually related with this. Now we have actually focused on this one-dimensional DNA. All this problem is actually dependent on how what is the concentration of that bead present on this DNA lattices. So we can say if we are knowing what is the occupancy index means how much of a particular biomolecule is present on lattice. We can average it out and we can know its concentration or its occupancy. So we have calculated actually occupation index. So these expressions have come after rigorous mathematics calculation. If somebody is willing to know that, if time permits, I can discuss it after the talk, means in the end of the slides, or after a talk also we can discuss. So right now you can take it from me that this O bar is actually representing average number of biomolecular species per microstate, and it is in the non-interacting regime. So this O bar represents a number of biomolecular species per microstate, and it has a relation with this lambda. Lambda is the absolute activity here, for example, in real system, and M is the number of DNA sites, where this Z is the grand partition function, and lambda has the same meaning. So now cell is not an example of, so these genetic systems are not an example of non-interacting frameworks. So there exist interactions, means genetic genetic interactions are there, chromatin interactions are there. So that's why we shifted towards nearest neighboring interaction, and we also calculated this average occupancy number. So which has the relation for this also. Rigorous mathematical calculation is there, which I've kept in the end, if somebody wishes to know. And from this, we have actually calculated fold change of expression. So what is fold change? So usually in the presence of RNA polymerase, whatever the expression we are getting, that we refers to as basal expression. And in the presence of transcription factor, now transcription factor can have multiple roles. For example, it can activate the expression also, and it can repress the expression also. So depending upon that, we can calculate the fold change, and where FA and FR has this basal component with it, plus some of the RNA polymerase that are interacting with the ligand or a transcription factor, those part also are linked here. So now, what is the total energy of our interacting system? Means what are the things which we are incorporating? So first of all, for example, we have one-dimensional lattice. On that, particles will be interacting with the DNA lattice. Those particles will be biomolecules. I'm referring to particles. Those biomolecules will be interacting with one another also due to the formation of as a long-range interaction, which we have called nearest neighboring interaction. And there will be a formation of DNA loop. Now it's like a tug of war going on between DNA loop and various biomolecular species. Those are coming near to one another because of the formation of DNA loop. So those things also we have considered. For example, so those species which are far apart, but because of this DNA loop, they have come close to one another. So they actually balances out one another, and that's how these three things we actually have considered. So now, what is our theoretical simulation details? We have considered grand canonical enzyme formalism to calculate this occupancy of transcription factor. It need not to be necessarily transcription factor. It can be any protein or biomolecule, or even ligand molecule also. And we have used a combinatorial approach because we wanted to simulate it also, so it fitted to our interest. So we have DNA as a 1D lattice of M equivalent, distinguishable, and independent site. So initially we started with indistinguishability, but DNA has specific and non-specific sites, so that's why distinguishability is coming into the picture. And here n number of proteins are interacting with the mDNA sites. So this is our Metropolis algorithm, where so this is simulation scheme where number of particles are going to be coming to the DNA lattices with the insertion rule by following this insertion rule, and we'll be deleting by following this deletion rule, and there can be displacement also from one DNA site to another DNA site. So now response curve for various genetic networks are presented. For example, we have considered various gene regulatory networks. So these are the simple gene regulatory networks we started with. For example, here we've considered RNA interaction with the DNA, and when they are in specific and non-specific regimes, and we have calculated average occupation index for them. And when this RNA polymerase will be actually triggered by this transcription factor, whether for activation or repression. And there could be oligomerization also between this among these transcription factors. And it can also be triggered by some other species which is coming from outside, which we are referring to as a ligand here. So we calculated here using this occupation index. So here we have, first thing we can see that these solid lines are well matching with the circles. Circle represents here the simulation results, and these solid lines are representing the theoretical results. And here we can see that as we are increasing the this absolute activity of RNA polymerase, we are getting the sigmoidal curve. And if we are facilitating the interaction of this RNA polymerase with DNA, for example, -8 kBt, so then it is getting saturated at higher rate. For example, in a red curve here, -8 kBt. And we also consider this, like I said, it is an cell is an example of interacting system. So we considered nearest neighboring interaction. And here we can, what we can observe, here also we observed, for example, for hump type thing, which is referring to the specific and non-specific states. And here we introduce nearest neighboring interaction. And here we can observe that there is a change in the slope of the curve, which is actually referred to the cooperative interactions. Those are existing in those large connected networks. So similarly, by varying transcription factors for these different cases, we actually have got the response function. So now we consider 180 lattices. Why 180? Because we varied from starting from 18, we went up to 1800. But what we figured out that 180 lattices works best for us. So that's why we considered 180 lattices. And like I said, various biomolecules are interacting with 1D lattices, and we have considered periodic boundary condition. Why? To remove edge effects. So now we wanted to see the role of this DNA looping along with the oligomerization of transcription factor and the ligand in regulating the expression. So we considered various networks, much more complicated networks. And here we have actually seen, we can see first we have considered transcription factor and RNAP, dimeric transcription factor with RNAP, ligand transcription factor and RNAP, and oligomerization of transcription factor in presence of ligand. And this is in the regime of in absence of DNA loop, and this is in presence of DNA loop. So these are the various species, those are actually existing for us. Which are coming for these various networks. So this is coming on linear segment. A linear segment means which are not part of the loop. So, and these are the various loop assisted species which are actually forming. So now I will show you the video. This is the simulation video. Which actually shows how these various species are coming and going and is responsible for forming DNA loops. So all those things actually, these things we have figured out using MATLAB code. And then we calculated as a varying function of activity of ligand, we calculated fraction for these species. And here this solid line represents in presence of in absence of loop, and dash line represents in presence of loop. So we saw that loop with loop and without loop actually controls this species on a linear segment where it decreases in presence of loop. And then we calculated a normalized fold change and dose response for these curves, where we have seen that when this fold change, when this transcription factor behaves as an activator, so then it increases expression, and when it behaves as a repressor, so then it well decreases the expression for us. And also ligand also showed similar type of effect. And also we have seen that when oligomerization interaction energy means transcription factor interaction energy is made more favorable, so then those it is getting saturated at lesser value of transcription factor, so which is expected, and similar is happening with ligand also. So then we concluded DNA looping and transcription factor and ligand interaction affects complex population. So then we wish to know that what is going to happen to the species those are forming due to this loop formation. So then as we move from left to right, so we can see that ligand species are decreasing here. So here we can see that the population of this species is varying from 0 to 0.2 is actually decreasing. So it clearly tells us that how this ligand species, when it is decreasing, concentration of ligand is decreasing, it actually is responsible for controlling the expression, means it is decreasing the expression. And if we move from top to bottom, so then we can see oligomerization of this transcription factor is decreasing here, and this also actually well controls the, means it also well decreases the expression for us. And apart from that, we can also see here we have varied this Hookean spring constant of DNA and epsilon Tn, and we can see that as the stiffness of the DNA increases, means the scale when it increases, so then a DNA is no longer able to form the loop, so then stiffness of DNA increases. So then if stiffness of DNA increases, so then complexes are not appearing after certain values. So like I said, there is a tug of war between the DNA loop and those species which are actually responsible for bringing the DNA close together. So that's why only certain values, fraction of values are appearing, and that too very smaller value of spring constant of DNA. So then we wanted to apply our model on bacterial system. So then we choose this lac operon system. Lac operon is a system which actually forms the lactose when glucose is no more available to it. So then it has this operon has 3 operators: operator 3, operator 1, and operator 2. And operator 1 is called main operator. Others are called auxiliary operator because it is actually majorly responsible for controlling the for repressing this gene. So here CRP binding site and RNA polymerase binding site. So this is the CRP molecule which binds on this site and is actually and it behaves as a activator of this RNA polymerase. Whereas this when this lac repressor binds, it binds as a tetrameric unit. So then when it binds, it actually represses this gene and RNA polymerase is no more able to bind. But when IPTG comes, so then it actually binds to this lac repressor and it is removed from this operator sites, and now RNA polymerase can bind and expression can go on. So these are the various promoter partition function which we have written for this case, which are for the activator and for the repressor part. And these expressions also have emerged out after mathematical calculations. So if somebody is interested, we can discuss it. So then these are the various different microstates that are originating for this lac operon case. So majorly O3, O1, and O1 with O2 and O1 with O3 are the major contributor which actually represses this promoter. And we actually also validated our result with the pre-existing experimental results in literature, where we have found that there exists a good amount of match between what our simulation results are saying and what experimental results are saying, though some little bit deviations are there. And these are in non-interacting regime. So then we actually try to give prediction for the interacting framework of this case. So where what we have done, we have actually considered nearest neighboring interactions. And what we have figured out here that when we are increasing lac repressor concentration, so then it is as we know that it is a repressor, so then it will bring the expression down. But when we increase the concentration of IPTG, so then we did not find major change in the repression curves. But when we actually included when we vary the CRP and CAM concentration when we increases, so then we have seen that higher activation function we have got for higher value of this CAM concentration. CAM is a ligand here, and here we can see that without loop and with loop case, here DNA is more stiff, here DNA is not stiff. So then we can see here that even though IPTG concentration is low, still expression is high. So then we did a literature survey and we figured out that this is called the leaky effect, which actually we were able to show with our simulation scheme. And leaky effect means even though IPTG concentration is less, still some of the leakage will be there in the promoter. And these solid lines are actually representing this interacting regime, and this not means these blurry lines are representing our non-interacting case, and the solid lines inside this not dark color actually are representing our non-interacting regime. And here we have seen that there is again a good amount of match between our theoretical results and computational results, and previously we have seen a good amount of match between our simulation results and experimental results. So then we shifted towards various gene regulatory motifs. For example, simple activation and repression, which could be induced activation and repression also, for example, for this case. And there could be various feedback loops also, feedback loop, positive feedback, negative feedback, and fully negative feedback, and feed forward loops also. So these are the various loops. On autoregulatory is also there. So these are the various loops which actually keeps on repeating itself in a genetic circuit. So that's why they are of interest. So then here I have shown the results related to feed forward loop case. So this side, it is coherent feed forward loop and incoherent feed forward loop. Why coherent? Because if we see from GX to GA we are going, so then it is positive, 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 and this side also it is positive. But if we see here, this is positive, but this arrow is negative. So then when there is positive negative thing, so then it is called incoherent. And when there is means when they are well correlating one another, so then they are called coherent feed forward loops. And these the results also we have shown, and where we have found that there exists bistability region for coherent type 4 and incoherent type 4. So since it is a tipping point conference, so then we have seen that various papers related to gene expressions are are being published recently, where they are actually talking about the gene expression using this tipping point. So what takeaway message you can take from my side, that at genetic and cellular level, various papers are related to tipping points. But of my interest, I would like to know that, for example, a tumor has started in one organ. How using this early warning signal, we can say that this tumor from one organ is actually going to spread to now another organ. Means what are those early warning signals which can tell us that now it is the stage that this tumor from one organ can go to another organ. So those kind of study I'm interested in. That's why I'm here. So what are the conclusions? So we have successfully developed a theoretical and simulation protocol for gene regulatory motifs. We found cooperative effect in nearest neighboring interaction, transcription factor ligand interaction, oligomerization, and they actually controls the expression that we have shown. And normalized fold change also actually significantly influenced by cooperative interaction. And we validated our model with the experimental results for the lac operon. And leaky effect also we observed. We actually predicted response function for the bacterial system in the interacting regime. And we have also done work with the network motifs. So these all findings, whichever are reported here, are coming from this paper. And with this, I'd like to say thanks to my supervisor, Dr. Sudipta, and I would like to acknowledge my institute, IIT Ropar, and UGC for funding. And I would like to end with this quote that essentially all models are wrong by George Box, but some are useful. Thank you.
Thank you. Questions? Any questions from students, faculty, anyone? Okay. If there are no questions, we thank Pankaj again for a very spirited talk. Then we will break for the tea now.